Good morning, everyone. Sorry, I don't speak Portuguese, Brazilian, but I hope you understand my English. I am actually from England, although I've worked most of my life in the United States. I want to talk about how I've been influenced by what is going on here in São Paulo in the laboratory of Edilamar and her students. We've had a good time. Now, in England, running is what football is to Brazil. And it began because we had the challenge of running a mile in under 4 minutes. And the first person to break that was a medical student from Oxford who in 1954 ran the mile in 3 minutes, 59 seconds, and so broke the barrier that had existed for a long time. So we love Roger Bannister, and he autographed my autograph book, so I'm showing that off this morning. With Edilamar, she came to my lab to study angiotensin, stayed with me for a year and a half as a postdoc, and I got very engaged in her model for exercise. So that's what I want to talk about today, and I want to talk about how that has led to very interesting discoveries. So my first paper on exercise, incidentally, was in people of what I thought was a very old age, but now I've reached that age. So it was in 1991. That was where we first really got good data on reducing hypertension by exercise. And all of these big study we did on older people who had hypertension, the hypertension went down when you gave them a series of exercise things to do. Edilamar Oliveira, Professor Oliveira, came to my lab to study stem cells, but at the same time, we started to realize that microRNA were really essential and very interesting non-coding non-coding RNAs that act really like gene switches. And ever since then, I've been interested in gene switches. She worked in the lab, and she is a real hard worker and a hands-on scientist. It's very nice to have somebody like that. And so she started to get interested in microRNA in the heart. And at that time, there were very, very few studies on microRNA in the heart. There was just one group that were in Texas that were doing very good. And I know you can't see all the details of this figure, but it's basically a group with Olsen and Van Ruij, and they mapped out some of the areas where microRNA are very important. And they did wonderful work on the heart, and that gave us the interest, because we were working on hypertension and cardiology, to look at microRNAs, and we're still doing that. MicroRNAs, of course, are as probably most of you in the audience know, they are a short sequence of 22 nucleotides, and in that sequence, there's an area called a seed. It's a matching area, a small number, 6 nucleotides, but if they match, then the microRNA works on that particular gene. That becomes the target gene for that microRNA. And the microRNA is mostly effective in suppressing genes. So it's like switching off a gene or holding a gene off until it's needed. Obviously, in embryonics, it's very important because if you can switch off at certain stages, you can have normal development. So microRNAs are extremely important in the embryo. They also, as you're here today, they become very important in changing the way cells behave, like cancer cells. And because they can switch off cancer cells, there's a big hope that microRNAs might be used to deal with cancer. Would certainly be nice. Anyway, in Edilamar's lab, they have a unique model of swimming exercise. They make their subjects swim. Obviously, as sedentary don't swim, but they are exposed to water as a control. And there are two levels of intensity. There's low intensity, which is 10 weeks of training, pretty low grade exercise. Then there's T2, or high intensity, I'm sorry. Low intensity. It starts as low intensity and then becomes high intensity as they add a little weight to the tail to make it a little bit more difficult. And then those rat athletes get this extra training. So we'll be talking about T1 and T2 when we talk about her results. Now, I've had the pleasure of being a co-author of some papers, and I just thought for this talk I will briefly summarize the things that they've discovered using this, what I call the Brazilian model of exercise. I know there are obviously other forms of exercise, a running wheel and other ideas, but this swimming one, which they've explored for now, getting on 10 years, has really been productive for them and really taught us so much. So I'm going to now summarize some of the things that we've learned from this brilliant exercise model. This is a paper in hypertension with the group, and this one was on the effects on the renin-angiotensin system. This is an interest of mine. I discovered a brain renin-angiotensin system some years ago, and we wanted to see if exercise affected it. And in this paper, we actually publish data on how it was affected, particularly the regulation of angiotensin-converting enzyme, and also whether angiotensin was raised by exercise or not. So what we learned from that is that left ventricular hypertrophy, which in these rats that were being trained, they develop left ventricular hypertrophy. We call that the athlete's heart. It's not pathological. There's a big distinction between physiological left ventricular hypertrophy and non-physiological. And even in a moment, I'll show you some data that we think we know what the difference is and what it's due to and which microRNA is most important. So in this heart, in T1, remember that's the low intensity, that was increased 13% above sedentary. And in the T2, those are our elite rats who got the more extensive training. It was 27% increase in left ventricular hypertrophy. So that was a good discovery. Now, was this due to the cardiac renin-angiotensin system? So we explored that and looked at the microRNAs that were in the heart of these animals. And what we found was microRNA 27A and 27B, which were associated with a decrease in angiotensin-converting enzyme, whereas microRNA 143 was decreased and associated with angiotensin-converting enzyme 2, which is a vasodilator, and angiotensin 1 to 7, which is also a vasodilator. So there were opposite effects of 27A and B and microRNA 143. Angiotensin was low in these rats, but angiotensin type 1 receptors in the heart were increased. So that was interesting in that paper. Next paper, exercise training prevents microvascular rarefaction. Rarefaction is the loss of blood vessels, mostly capillaries. Now, hypertension is due to increased resistance of the blood vessels. And if you have fewer blood vessels, the resistance goes up. We found in this study that exercise training reduced high blood pressure in rats, as we had found also in humans, it increased the levels of microRNA 16, 21, and 126. Increasing these microRNAs corrected for capillary rarefaction and revealed molecular mechanisms for vascular health, both pro-apoptotic and anti-apoptotic, that needs to be balanced. So this was an important paper in telling us how the hypertrophy is controlled by microRNAs. Now, rarefaction is really a very interesting area. It's been known for some time, and it's the blood vessels just basically start to disappear, and that increases blood flow resistance. It has been described in patients with early stage of hypertension, so it's a very good reason to think that rarefaction is a cause of hypertension. And if you could stop rarefaction, you could stop hypertension. That would be a hope. Decreased vascularity leads to inadequate supply of oxygen, and rarefaction increases total peripheral resistance, and I expect most of the students know this simple formula: blood pressure equals total peripheral resistance times cardiac output. Now, in a recent paper, this has been confirmed with obese rats, and in obesity, microRNA 126 induces capillary rarefaction, this paper is it's just coming out, so I don't have all the details here. But anyway, it shows that this is another finding from this exercise model. This is another paper on the aerobic training promotes physiological cardiac remodeling. As I said, left ventricular hypertrophy normally is a bad sign. It's a sign of pathology. But if you're an athlete, if you're training, it's one of those byproducts of training that your heart grows bigger. It's a muscle, so it grows bigger. And you have to differentiate between those two. Normally, if you go to a doctor's office and they do an X-ray and you have left ventricular hypertrophy, the doctor's going to be very worried. But if you're an athlete, that's usually a good explanation. So in this paper, we summarize a set of microRNA. Some have anti-hypertrophic effects. These include microRNA 1 and 133, which are by far the most commonly found in the heart, and then 26, 9, 98, 29, 378. I hope that some of you in the audience are working and then you see your favorite number. Please tell me later if you find it. We're talking about your favorite microRNA. The pro-hypertrophic ones, those that increase, go with hypertrophy, include these numbers, and I won't go through them all, but they are a substantial number, of course. And then there are groups of microRNA that we consider to be cardiac specific. So they're called myomeres, being in the muscles of the heart. As I mentioned, 1 and 133, both A and B, 208, A and B, and 499. Just 4 of them really are specific for the heart. And then one that was discovered here is the effect of microRNA 29 AC. We'll come to that in a moment, and it's anti-fibrotic. There's another, well, this is the paper. It's by Ursula Sochi in Dr. Oliveira's lab, and it actually shows that a bad heart is a heart that has a lot of fibers in it. The fibers are there because of the collagen gene, and microRNA 29 inhibits the collagen gene. So in these athletic hearts, these left ventricular hypertrophy of athletes, they don't have fibers, they don't have fibrosis. And that's a very big distinction. Their hearts are just pure increased muscle. So that was a big discovery, and I think it's a very important one. Normally, a heart is like this, then it can either become a physiological hypertrophy or a pathological hypertrophy. They're roughly the same size, they look the same on an X-ray, but in fact they're entirely different because it's full of fibers due to collagen genes. And this is not, this is all muscle. To explain that in more detail, there's a whole process that actually involves angiotensin and aldosterone and fibrosis of the heart, which in pathological left ventricular hypertrophy is a very significant problem because the collagen fibers that build up, break up, and actually ruin the heart. And this could lead to heart failure, and invariably does. So it is a very bad thing to have if you're not an athlete, if you get this. So just let me summarize there. Exercise increases microRNA 29A and 29C, which inhibit collagen 2 and 3. The collagen normally increases fibrosis, therefore microRNA reduces the fibrosis and reduces fibrosis in the heart, and therefore exercise leads to microRNA, which reduces fibrosis and gives you a healthy heart. It's a simple message, but it's very important. Now, I mentioned earlier the group in Texas, Olson and Van Ruyd, and they had a very nice summary of effects in Nature. Actually, this shows how fibrotic a heart can be. This is, of course, not with exercise. That's the contribution of Dr. Oliveira. But the heart normally gets fibrotic, and then it has all these microRNAs, which I'm sure you can't read from the back of the room, or maybe the front of the room. But this is a summary of the number of microRNAs that are involved in the heart. And we've added microRNA 29 and exactly how it works in this state. Now, I bring you up to one of the most recent papers published last year, and it's called Epigenetic Control of Exercise Training. Epigenetics are something that puzzle us, but they obviously have potential for study, for treatments and so on. Epigenetics is not genetics. It's over and above genetics. It's things that influence the way our genes work based on our experience in life. It's one of the ideas for a healthy life that you have good epigenetics. The things that serve you well, like exercise and good food, will have a positive epigenetic effect. So they're very important, and there's even hints that some epigenetics carry over into the next generation. I'm sure that's very debatable, but there are ideas that a healthy life leads to somebody else having a healthy life in your offspring. So this paper, which came out last year, is in Clinical Science, points out that microRNA 208 leads to a number of changes in what we call epigenetic genes. Most epigenetics is really the modification of DNA by methylation. DNA methylation is an epigenetic mechanism. It occurs by the addition of a methyl group to DNA. DNA methylation and also histone modification are epigenetic mechanisms. So either you have DNA methylation or you have histone acetylation, and then you are dealing with something that is not in the genome, but is an effect on whether the genes will be expressed or not. MicroRNAs regulate epigenetic genes and are regulated by them. It's a feedback. And what this study shows is that exercise has epigenetic effects. So you may not be born an athlete, but with training you can become an athlete, and at the same time, because of this epigenetic effect, you can have a healthy heart. So here's the model, here's the rat in training, and it's going to inhibit microRNA 208A and 208B. That inhibition releases a number of epigenetic genes. We've got MED13, pure beta SOC6, and just a string of them that actually work on myosin heavy chain. And that myosin heavy chain is what makes the muscle, particularly the cardiac muscle, a muscle. But it's a balance between alpha MHC and beta MHC. So one is positive and the other is negative, and what you're trying to do is keep a balance. And what has been found in this study is that 208 is a controlling microRNA for all these epigenetic effects. So this is new territory. It's a new idea to start looking at microRNAs for epigenetic effects. Epigenetics seems fairly complicated, but if you just think of histones and DNA methylation, they both work together. The histones, of course, in the chromosomes, the chromatin is made up of the histones and the DNA wrapping around those. And if at some point you have methylated DNA, that is going to stop those genes from activating. So if you stop the genes from activating, if they're bad genes or genes that will lead to something like fibrosis, then you are going to have a good effect. And it's very simple chemistry based on cytosine being methylated in the DNA, or a little more complicated with the histones and deacetylation. But that is the basic mechanism, and that's the mechanism that we're now beginning to find is altered by microRNAs. A little bit more from the chromosome. When the chromosomes, everything is all tightly packed, and if you unpack it, you find it's a mass of histones, DNA wrapped around, and they can be either well-spaced or they can be tightly spaced. And if they're tightly spaced, they might switch off the message, the DNA or the gene will not be expressed, and if they're widely spaced, they might be switched on. So this structural change is very important, and if you can control it with something as simple as a microRNA, you're really controlling the expression of genes. So in a summary, this comes from NIH, the summary is that these two mechanisms, methylation and the acetylation of histones, are ones that will affect whole gene expression. And what we're beginning to realize is that they can be controlled by microRNAs. We've looked at 2008, but others have now reported microRNA 9, 34, 124, 127, 137, 148, and so on. So all of those are regulating the epigenetic expression of DNA methylation and histone modification. So I think it's new and exciting, and I just summarize by what has been found with this Brazilian training method. It has opened up a lot of interest in exercise, in microRNA 29C, as I've just been mentioning. Others, like microRNA 27A and B, affect the angiotensin converting enzyme and therefore vascular resistance. A decrease in 143 affects ACE2, which is increased in hypertension, and also causes vasodilation, so it's kind of fighting hypertension, but it's something we are looking at. And then microRNA 208, which affects this is thyroid receptors, which are in the heart. It's the balance of alpha and beta myosin heavy chain expression that it's controlling. And in hypertension, that's increased too. So I'm going to close here with my conclusions. From a simple model of exercise, the swimming training model, T1 and T2, has come a number of discoveries of how exercise improves heart function and reduces hypertension. The Brazil group have extended and enriched their findings by elegantly following the molecular changes with exercise, most notably with the identification of various microRNAs and their activity. Their extensive knowledge has led to their model contributing to a new field of research in exercise, the science of epigenetics of exercise. And with that, I thank you for your attention.